So you get some practice for that. It's kind of fun. And then I also want to do what's called the chi-square test. We haven't done that in here, right? I teach too many years. I forget what year I've done it. One year I haven't. Chi-square is a good time. This is a good time to bring it up because of genetics, because it's a statistical test that we use to determine significance of various types of results, often genetic results. All right, so let's get to the notes. We left off talking about recessive traits and how it's not easy to get them. You have to have 2 copies. They can result in some pretty nasty conditions. And there's lots of them. We're not going to do them all, obviously, but just a couple to illustrate a couple. One is cystic fibrosis. Cystic fibrosis is common in people of European descent. And it results in a chloride channel in the cell that is not working. And because it's not working, it causes the chloride ions to build up outside of the cell. And that draws water out of the cells. It makes the mucus a problem. And so you find in a lot of organs of people with CF, you find a buildup of mucus, especially in the lungs. And that can lead to breathing problems. It can lead to easily gotten pneumonia. People with CF get sick very easily. And it usually leads to a shortened life expectancy because of that. Because in order to get rid of that excess mucus, right now, the only way to do that is to suck it out. So you have to aspirate it out. And doing that causes damage to the lung tissue. And that damage to the lung tissue, along with prolonged, bacterial infections and such, usually leads to a shortened lifespan.
Wait, it affects 1 out of 2 people?
Yeah, of European descent.
So it's like something common.
It's fairly common. We usually have in the school a couple of kids with CF. And as teachers, one of the things that we are required to do when that happens is we're required to wipe down the desks before they come to class and after they come to class. Because again, getting sick is a really big thing. So making sure that everything is as clean as possible is a huge deal for people with cystic fibrosis. See if it'll move. All right, how about this one? Will that move? There we go. Another recessive disease is sickle cell anemia. This is even more prevalent in populations who are from African American descent. 1 out of about 400. And this is one of those where there's just one change in a protein sequence. So 1 letter change makes 1 amino acid different in the protein. And what it ends up doing is it ends up making your red blood cells, which are supposed to be nice and round and smooth, it makes them pretty jagged and pointy, kind of looking like a crescent moon. Or a sickle. In old days, they used to use a sickle to get at the wheat in the fields. They would use that to cut it down, a scythe. And that's where that sickle comes from. This means that red blood cells' job is to carry oxygen to your tissues. If it is long and skinny, it doesn't carry oxygen very well. And it tends to get jammed or stuck in the blood vessels. And so you can get a jam of these red blood cells that back up, and that can cause a lack of blood flow to a certain area. And if that's to a vital organ, that could be a real problem, a real crisis for the person. We're getting better at treating people with sickle cell. So they still have a shortened life expectancy, but much longer than they used to. The weird thing about people with sickle cell is, well, I don't know if it's weird. So you have to be two small S's to have sickle cell. But this thing is really slow today. If you are heterozygous, sometimes you have a few of the symptoms, not much, but you may have a few symptoms if you're heterozygous. But over time, you would think because of their shortened life expectancy, you expect people you would expect this allele to become less and less common because it isn't helpful in survival. It isn't helpful in reproducing and having children. But it turns out that about 1 in 10, this is huge, African Americans have the sickle cell trait. In other words, they are carriers for it. Okay. That's really high. You would expect that to be a lower number, again, because over time, we haven't gotten to evolution yet, but over time what happens is because that allele is not beneficial, it's actually detrimental to somebody's health, you expect fewer and fewer people to have it. But what is interesting, and they figured out why it's such a high rate. People who are heterozygous for sickle cell anemia are much less likely to get malaria. They're protected against getting malaria, which can also be life-shortening. Remember, I don't know if you remember, malaria is caused by a parasite that is carried by mosquitoes. So when you get bitten by a mosquito carrying the malaria protist, it infects you, and then you get the malaria from that.
Wait, how does having like the recessive
I don't know what there must be some conference that it gives you, like maybe it makes it harder for the malaria to infect cells.
Because the blood is like.
Yeah, I don't know if it's less surface area, if it's just something simple as that. Maybe it makes it harder. But the other thing is you only find this. So you find this 1 out of 10 African Americans, but it's from a specific area of the continent of Africa because that's where malaria is prevalent. You don't find people having as much sickle cell if you go outside of that particular band, that particular area. And I always look for this picture and always, let's see, sickle, Africa, Africa continent. Continent image. Wow, my computer's slow today. Come on. Just show it to me. Just go. There we go. Oh, good God. Can't I just see the picture? All right. So this is always what happens when I don't actually think ahead. So if you look at this picture over here on the right, this is where you see the greatest amount of sickle cell. And that's also where you see the greatest cases of malaria. Outside of those bands, see how it gets lighter colored? there you see less sickle cell and you also see less malaria. So having that sickle, being heterozygote for it means that you are protected against malaria. But going back to our PowerPoint, if I've got two parents who are, did it again, who are big S, small s, heterozygous, how many of their kids are going to have sickle cell? Yeah, 1/4, 25% of their kids will have it. Okay, so it protects them, not so good necessarily for their children. Okay, so it's just kind of an interesting, study in evolution and how things, they stick around for a reason. In this case, the sickle cell sticks around because it does help in survival in certain areas of the world. This is just showing you the different proteins that are made and how you result in that sickle cell shaped red blood cell as opposed to the nice round and smooth one. Those are all recessive disorders. There are some, and you should know these recessive disorders. In other words, if they might say cystic fibrosis, and you just need to know that's a recessively inherited disease. There are also some that are dominantly inherited, and dominantly inherited ones don't occur very often. They sometimes arise by mutation, so they just appear out of nowhere. A good example or a common example of that is dwarfism, or I can't say this word, so achondroplasia. Chondroplasia is a dominant trait. You only need one allele in that case because it's dominant. Dominant's going to show, so that dwarfism is going to show. But if you had two parents who are, in this case, we're showing one parent that has the disorder and the other one has a normal phenotype. You're going to have half your kids showing the dominant achondroplasia and half showing the normal phenotype. But you could also have two parents who both have the condition. And you could end up with a quarter of your kids having the normal phenotype. Just depends on what you are genetically, genotype. Mm-hmm.
Like they know that.
Yeah. Yeah, which is why. Yeah, another one. Which is why genetic counseling can be really useful for some people if they think that's in their background, or the cystic fibrosis or the sickle cell. To decide, do I want to have kids or maybe I want to adopt instead, because, that's awesome and you still get children and you don't have to worry about, a large percentage of them being exposed to some, deadly disease or at least life-altering disease. Another one that you should know about is Huntington's disease. Huntington's is a mental degeneration. It's a degeneration of the brain. And the really irritating thing about this is it shows up usually or often after you have had kids. So it's dominant and it is fatal. But it usually results in a fatality after the person has had kids. So they've already potentially passed on to their kids.
What?
And their kids don't know they've got it or not unless they get genetic testing. Yeah.
I mean, like right after they have kids, they're just like.
No, it's not. They're not related.
Okay, so
But you might have kids when you're 30, and then when you hit 40, it's like, oh crap, I've got Huntington's. My kids are 10 years old, 10 and 12 years old, and they might have it or they might not. They don't know. You can check. There is a test for Huntington's. A lot of people don't want to know. Memory goes with Huntington's as well as your motor control. of your muscles. So a lot of people, they forget their keys one day. They don't want to know, well, maybe that's just because Huntington's starting. Or are they just normal people who forget that their keys,
Wait, so it like
Uh-huh.
is it because like your age you just get older and then it happens, or
Mhm.
is it really just because you have kids and then have
No, it has nothing to do with having children.
Okay. I was like
Yeah.
that'd be
You get
crazy.
Huntington's with or without kids.
Okay.
It's just that if you have kids,
You
you've
would-
already had them, and you've already potentially passed it on and didn't know it. I think we already talked about this, the idea of multifactorial inheritance, meaning that there's so many diseases now that are not just genetic. And so we can find a genetic link, but that doesn't mean that it is just genetic. A lot of it has to do with lifestyle, environmental conditions. So alcoholism, heart disease, cancer, mental illness, both of those have both a genetic, or all those have a genetic and an environmental component. So no matter what your genotype, our lifestyle has a tremendous impact on our phenotype, what we're actually going to physically end up showing. So eat your veggies, boys and girls.
And I was like, wait, what about
Genetic counseling can be very helpful to develop a family history and to make predictions about what potential things your kids might be exposed to before you ever, have kids. So it allows you to make a pretty good decision whether or not you should have children. So a genetic counselor can determine the risk a couple can have of having a particular disease. Again, that doesn't mean they're going to have it, right? If it's a 25% chance, you flip a coin. Maybe, maybe not. There are some tests that can be done in utero. So after the baby has been conceived. Come on. How many times do I have to hit this? God, this is slow today. Come on. All right. Amniocentesis. This is where liquid is removed from around the fetus because a fetus, just like us, shed skin cells. And those skin cells will be present in the in the fluid surrounding the baby. And you can remove that. You can extract out those skin cells. You can look at the DNA, and you can do that karyotype that I talked about. So that you can look at, do they have the right number of chromosomes? Is there any kind of abnormality with regards to that? There's also chorionic villus sampling, which takes takes a few cells off of the placenta. And then, of course, there's also like ultrasounds that can be done to look at the health of the child in utero.
Is amniocentesis done for everyone, or is it just?
It's not done very often because there is a slight risk to the fetus. So you want to try and minimize that if you can.
Literally take it out. Okay.
You take fluid out.
I was like, what?
But it's a fairly substantial needle that they stick into you to get at the baby, and they do it in conjunction with ultrasound. So they can see where the needle is compared to the fetus, but still not a great thing to do. Ooh, that's a good question. I'm not really sure.
Is it soon or when
It's usually later on. Yeah. But I don't know exactly how long on. How far along? Throwing Tufts under the bus, are we? Okay. There are some diseases that babies are screened for right after they're born, that are fairly straightforward, that are done just as a matter of routine now. Because if they have certain disorders, and it's not apparent right away, you don't test for it, the child could have some really bad implications for the kid. One is called a test for PKU, or a phenylketonuria. And this is the inability to digest the amino acid. So you have 20 different amino acids. The amino acid phenylalanine. And phenylalanine is in different products that an infant might take, might ingest, and it causes permanent brain damage because you can't break that amino acid down, and it accumulates in the brain and causes damage to the brain. But you can do this really common test and find out if the kid has PKU. And through diet, you can keep those foods out of their diet, and they're, it's pretty inconsequential if you know about it. Yeah.
So does that mean like when they're an adult, they can't have like chicken or anything?
So one of the things that sometimes happens is they actually, quote, grow out of it. They grow out of the disease. Let's see. I can't remember. I know one of them, which is not something you're going to feed your infant, is diet soda.
Why would you feed your?
I just said you wouldn't, but you 3 or 4 year old, you want, mommy, can I have a sip of your Diet Coke? And no, you can't. You'll get brain damage. Foods to avoid. Yeah. Okay. Oh boy. Oh, so it's all. You're right, Nolan. It is chicken. Yeah. It's a lot of things. Yeah. I mean, the dairy's the real problem, right? Because you'd have to do dairy-free milk somehow.
So you don't want protein?
Yeah.
So you can't wait. Yeah. What protein?
How do you like?
Soy? Soy, maybe?
Stay healthy?
Can you eat bananas? Yes. Many fruits and veggies. It actually doesn't say bananas.
It does.
Yeah, it does.
Second thing right after that banana.
Oh, yeah. Sorry, my bad. Yeah, berries, mangoes. Okay. Okay.
Wait, can you have fish? You can't.
Oh, fish is in there too. I mean, I'm sure that this is, I wouldn't use Google as my goal.
Primary.
If
I think it's okay.
I would think it's okay. Let's find out. Yeah, no.
That's Pinterest.
Yeah, Pinterest. But it's from the PKU clinic in the University of Washington. That's just if you want to save it. Anyway. Yeah. But that's one of the tests that they do to make sure that, because it does happen. It's fairly common. Don't want that to happen. Okay, so we're going to move out of chapter 14. And we're going to move into chapter 15, which is it's just a continuation of genetics, really. For you all, it's not really a big deal that it's a different chapter because it's more genetics. But Mendel was pretty clever. I mean, he came up with some spot-on ideas without knowing about chromosomes, without knowing about genes. He called them factors, but we know that those factors are actually genes. We know that they're located on chromosomes. Chromosomes get passed down. We didn't know any of that at the time that he was doing all this. And a couple of things that he basically predicted was the law of segregation, which says that if you got two alleles, they separate from each other during meiosis. So if you have sickle cell, let's say you're a carrier for sickle cell, you have a dominant trait that's free of the sickle problem. And you have one that's recessive that has the sickle cell trait. But those two separate from each other during meiosis. There's also the law of independent assortment, which says that if you're looking at genes that are on non-homologous chromosomes, they're also going to separate from each other. So you don't necessarily see two genes, even though they might be together in a parent. The offspring might show one of those traits, but not the other of those traits, because those, if they're on non-homologous chromosomes, they will separate from each other, and so those traits will get they'll get sorted independently of each other, and they'll come up on different chromosomes, and you can end up with completely different looking kids because those things are not they're not joined together. We're gonna start by talking about Morgan, and I'm not gonna spend too much time talking about him, and you don't need to remember his name. But Morgan's big deal was, and if you do like genetics and you decide to study genetics, I got either good or bad news depending on whether you like them or not. You're probably going to study, and if you study bio in college, you'll probably study this as well. You get to study fruit flies. Hey. Oh. Fruit flies that we use in the lab are called Drosophila melanogaster. More commonly, they're just called Drosophila. Yeah, so rather than call them fruit flies, they call them Drosophila. It's just a very common name. Morgan worked with these guys. This is around the turn of the century, so 1900s. The good thing about fruit flies is, and I've worked with fruit flies, I've actually had students doing independent projects with fruit flies. Turns out that fruit flies are they do not like blue light at all. It like totally messes with their reproductive systems. Anyway, Drosophila produce a lot of offspring in a very short period of time. Every 2 weeks is an entire life cycle for them. And they only have 4 pairs of chromosomes. So they're pretty simple to study. You can look at the genetics very easily. And there have been tons of different strains of Drosophila that have been developed. Ones that are blind, ones that have mutant-colored eyes, ones that have that don't have the ability to fly. Has anyone gone to PetSmart and picked up fruit flies?
No.
For their gecko or?
Unfortunately,
Oh shoot! What is it? Oh, is it a cockroach? All right, we're back. Sorry, I had to get rid of Charlie. Charlie was a little American cockroach. It's kind of messing with people, so we got rid of him. It's all good. Okay, so back to fruit flies and not roaches. So Morgan noted that there was what he called a normal phenotype for fruit flies, or a quote wild type, which is just meaning that that's what you really saw in the wild. But that he also found that occasionally flies were born not with red eyes, but instead they had white eyes. So they were a mutation that resulted in no pigment being produced and white eyes as a result. And he started working with these fruit flies, and fruit flies are very easy to breed. Like I said, very easy to work with and kind of fun, I think. Personally, I know I'm kind of alone in that. In one experiment, Morgan mated male flies with he mated male flies with white mutants and female flies with red eyes with wild type. Okay, so he was taking white-eyed males, mutants, and females that had red eyes, which were the wild type, and he crossed them. And when he crossed them, he found what you always find, or what we had traditionally found, which was all of them had red eyes in the first generation. Oh yeah, that makes sense. But then he bred the second generation. And when he did the second generation, he found an interesting thing, which was he found the 3-to-1 ratio of red to white eye, but it was only in the males that you found white eyes. So only the males had white eyes, not the females. It's like, okay, well, does that mean females can't get it? And he did some more testing, and from all of his work, he realized that that there are some traits that are found on the sex chromosomes that code for, in this case, eye color. Okay. So first generation, they all had red eyes. But then in the F2 generation, he found that many of the males had white eyes. And there we go. Morgan's discovery that a trait correlated with the sex of the flies was key in the development of the idea that there are these things called chromosomes, and they get passed down. And so whatever is on them, because now we're seeing, okay, well, males and females have this different inheritance. Why is that? We know now that humans and other mammals have two types of sex chromosomes. And these are, for the most part, code for traits that are associated with biological sex. And there is one large or larger X chromosome, and there's one smaller Y chromosome. A person who has two X's is going to develop as biologically as a female, whereas individuals who have an X and a Y will develop into biological males. And I'm using biological because that is different from one's gender identity, so that we're still kind of grasping with how that plays out in terms of biology. We still don't really know that. We do know that people definitely don't necessarily identify with the gender that they were assigned at birth, right? You could be XY and feel completely female, or vice versa. So that's a different argument from what we're talking about. We're talking about organs that are developing, uterus versus the prostate and internal of biological males versus females. So that's what an X actually looks like, and that's what a Y looks like. There's not a lot of chromosomes on the Y. A lot of genes on the Y. I think there's like 40 or 50 at last count. Short segments at the end of the Y chromosome are homologous to the X, allowing the two to behave like homologous chromosomes during meiosis. There is a gene that's located on the Y chromosome called the SRY gene. And that SRY stands for sex determining region on the Y. And it's responsible for if the SRY gene is turned on, if it's obviously present on the Y and turned on, the embryo will develop testes. Okay? Develop testes if that SRY gene is there and gets turned on. And if it doesn't, then you will instead have the ovaries being produced as a result. I'm over time. Darn it, Charlie. Okay. Didn't I just click on this? All right. Let me do this slide quick and I'll stop after this. A gene that is located on either sex chromosome is called sex-linked because it's linked to either the X or the Y. Genes on the Y chromosome are called Y-linked. Genes on the X are called X-linked. There's only, it's more than 40. There's 78 genes that code for 25 proteins on the human Y chromosome. Genes on the X chromosome is around 1,100 of those. And you don't need to remember the numbers. It's just the idea that there's very few on the Y compared to the X.
Does that mean boys have less traits overall?
No. They're just not on. They're not on the Y chromosome. All right. So when we return tomorrow, what I will do is I will talk about sex-linked traits, and I'll do some sex-linked trait problems.